My group really focuses on understanding fibrotic diseases. This is a class of diseases that can affect really any major organ. And the underlying disease that causes fibrosis varies quite a lot, even in the same organ. But at the heart of organ dysfunction is formation of this collagen-rich scar. So the scar tissue, this fibrosis, really disrupts organ function and eventually leads to organ failure. And this is true across these organs, across these diseases. And it's not really the problem that the scar tissue develops in the first place, but it's really that it can't be degraded. And despite this major effort in developing pharmaceuticals that can reverse fibrosis, we really have not been successful. And that's because we don't really understand the biochemical features that dictate scar degradation. So my lab has set out to answer this question, and we started in the heart, where an injury like a myocardial infarction or heart attack results in this massive cardiomyocyte death event. And the body treats that like it treats any other wound. It starts removing the dead cells and depositing scar tissue. And the scar tissue deposition, like in any other organ, is really harmful because it can't do what the cells of the organ can do. It can't move, it can't function the same way in the heart, it can't pump. And so that actually leads to further remodeling and eventually leads to heart failure. In the heart, this is true for all adult mammals, but in the zebrafish, the same sort of injury that leads to a formation of scar also leads to formation of scar tissue. But unlike us, that scar tissue can be completely degraded as the myocardium regenerates. And in both cases, that scar tissue is made up of the same stuff. It's collagen rich, and collagen is really the protein that gives the scar tissue its structure. Despite these similarities, though, in one case the scar is permanent, and in the other case the scar is resorbed. And the idea had been that highly cross-linked collagen is what determines whether or not scar is degraded. And those collagen cross-links is really what the bonds that form between collagen that gives it its stability. So we set out to answer this question, is degradable collagen less cross-linked? We know quite a lot about how collagen cross-links, and it really starts out with the oxidation of collagen lysine groups to form these aldehydes. And these aldehydes are really crucial for cross-linking. They are the reason that collagen cross-linking is initiated because they can react with each other or with lysine groups on adjacent collagens to form these initial cross-links that can undergo further modification as the scar stabilizes. But the key here is this is the only enzymatic step in the collagen cross-linking cascade. And so we can develop tools that bind specifically to these L-lysine aldehydes and accumulate on tissue that's undergoing active collagen cross-linking. So in this case, we developed fluorescence imaging probes, but we can really develop any image. We can tag these functional groups with any imaging reporter. But here we use fluorescence because the zebrafish heart is so small and we needed something that would give us high spatial resolution. So of course, I don't have time to go through the probe development with you, but suffice to say that we do a lot of validation and optimization of the probes, and we use our optimal construct in these studies, which is the probe we called TMRO. So TMRO, one thing that I should mention is that it binds to healthy tissue that's also rich in collagen and elastin. So the structure is the aorta. So the healthy aorta is rich in oxidized collagen, and so our probe can bind to that tissue as well in the healthy animals. But what we saw when we injected infarcted animals with our probe is that at early time points after injury, where collagen hasn't really started being deposited, we don't see much accumulation of our probe. That quickly changes and it peaks around 21 days after injury. So collagen cross-linking peaks around 21 days after injury. And it decreases, and the decrease after that is not a result of lack of collagen cross-linking. It's just that scar tissue is maturing. So there's areas where there's clearly scar tissue, but that scar tissue doesn't have those aldehydes because those aldehydes have already been consumed and have undergone cross-linking. So we observed here is that collagen cross-linking is robust even 2 months after injury in the case of the mice, and there's evidence for scar maturation. Now, in the case of the zebrafish, we use the zebrafish line that has cardiomyocyte-specific expression of GFP, so that allows us to delineate where the injury is. And similar to what I've shown you before, our probe TMRO binds to the bulbus. So this is a healthy structure that is rich in oxidized collagen. So our probe accumulates there as well. In the early days after injury, we don't see much accumulation in the injury area, but that changes quickly. And the story is actually pretty similar to what we see in the mice, where collagen cross-linking peaks around 21 days after injury. But unlike the mice, the zebrafish myocardium completely regenerates. So the zebrafish injury decreases over time. And then at 62 days after injury, the scar tissue is completely gone, and we don't see accumulation of our probe anywhere in the ventricle. Now, this really showed us that even though there's a lot of collagen cross-linking going on, that scar tissue is degraded. And so if we compare the peak collagen cross-linking in both species, it turns out they're just not that different between the zebrafish and mice. So that led us to ask, what happens to those early cross-links? Do they mature? We looked at this degradation-resistant collagen crosslink, PYD. This is the most well-known or well-studied called mature collagen crosslink. And what we found is that it's just completely absent in zebrafish infarct, even when we account for the amount of collagen. So it's completely absent in zebrafish infarct. It's highly abundant in the mouse scar. And it turns out that this is actually because of a key hydroxylation step that happens to collagen prior to the cross-linking starts. So collagen can be hydroxylated. The collagen lysine groups can be hydroxylated with lysyl hydroxylase 2. And whether or not, regardless of whether or not the lysine groups are hydroxylated, these lysine groups are oxidized and collagen cross-linking can occur. But if these groups are hydroxylated, then these mature collagen cross-links form. But if they're not hydroxylated, then those cross-links cannot form. And we've quantified the amount of these hydroxylated lysines. We've seen that they increase with mice in the mice infarct, but they don't at all change in the zebrafish infarct, and which really tells us that these mature collagen crosslinks are absent in the zebrafish infarct, and this is likely due to the absence of lysyl hydroxylase activity, and because the collagen on the zebrafish infarcts is just less hydroxylated. So now, of course, if the takeaway of this project is that it's not the extent of collagen cross-linking, but really the type of collagen cross-links that dictate the scar degradation. So in permanent scar, those collagen cross-links are locked into place, but in the reversible scar, those collagen cross-links are more or less open. And we've actually shown that this is not just true in the zebrafish heart, but this is true in other mammalian models of reversible fibrosis. Now, finally, I just want to take the last minute to talk about our efforts in translating some of these technologies. We're really interested in trying to stratify patients based on disease activity in fibrosis. And this is because a lot of fibrotic diseases have patient populations with very heterogeneous disease courses. So some patients progress very rapidly and some progress very slowly, or anywhere in between. And the big problem is that during clinical trial development, all of these patients are lumped together. So that dilutes out any effect that you would see because of treatment. And that results in the majority of clinical trials for fibrotic diseases failing because they can't show differences or drug efficacy, essentially. So there's really an urgent need for tools that can sensitively detect disease activity. So similar to what I've shown you before, we can target oxidized collagen because it only happens during active disease. We can develop these probes that we can tag with any reporter, but in this case, it's a PET reporter, so it's a radioactive gallium. And what I've shown is that we can use these probes to really sensitively detect changes in disease activity with imaging, non-invasive imaging, which we cannot do with the state-of-the-art high-resolution CT. So this is what's used clinically, but it just can't detect the changes in different states of disease. So we really think that developing this kind of technology could really de-risk clinical trials. So my program really focuses on developing tools that can enable understanding of disease mechanisms, identifying therapeutic targets, and clinical translation of these tools that will enable de-risking clinical trials. So thank you so much for listening, and here are my acknowledgements. Thank you.